that represents the light absorbing or the light absorbed at each wavelength by the pigment, that is called as the absorption spectrum. So we are studying the absorption spectrum here. And you see the curve showing the rate of the photosynthesis at different wavelength of light, that's called as action spectrum. So this is action spectrum. So you be clear with the absorption spectrum and action spectrum. So absorption spectrum, you see that the actual photochemical efficiency by the continuous line, and the dotted line shows you the absorption spectrum here, actual how much absorption is taking place, whereas here the action spectrum you could see the wavelength and the action spectrum. So you see here, this is the absorption of the light by the chlorophyll pigment, the graph showing the absorption spectrum of the chlorophyll A and B. So this is the chlorophyll A and this is the chlorophyll B and their absorption spectrum. And here this graph is showing the action spectrum of the photosynthesis, like the rate of the photosynthesis that's measured by oxygen release. So this is the action spectrum that is there for photosynthesis. Now, absorption spectrum is studied with the help of spectrophotometer, with the help of what light is being emitted by the substance in the spectrophotometer, which is a device we get to understand. So, the absorption of the spectrum of the chlorophyll A and chlorophyll B indicates that these pigments mainly absorb the blue and the red light between 430 nanometers. So they basically absorb the blue and red lights at 430 nanometer and 662 nanometer for the chlorophyll A specific, and 455 nanometer and 644 nanometer, that's the blue and red light respectively, 455 nanometer and 644 nanometer for specifically chlorophyll B. Rate of photosynthesis, absorption percentage, the action spectrum, the absorption of the spectrum of the chlorophyll A and B. Action spectrum shows that maximum photosynthesis takes place in the blue and the red region of the spectrum. Now, the first action spectrum of the photosynthesis was studied by T.W. Engelmann, 1882, using the green algae Spirogyra and oxygen-seeking bacteria.